Hello everyone, my name is Juan Sebastian Pino, PhD student in biology at the University of Antioquia. Thank you for joining me presentation. Today I'll be discussing our study on the comparative transcriptomic responses of human bronchial epithelial cells, specifically A549 cells exposed to diesel exhaust particles or DEPs. So, diesel exhaust particles are tiny particulate matter emitted from the combustion of diesel fuel, containing a complex mix of soot, organic compounds, metals, and other toxic substances. Due to their small size, they can penetrate deep into the lungs and even into the bloodstream, leading to a range of health issues. It's known that short-term effects of this exposure include headaches, irritation of the eyes, nose and throat, respiratory issues like coughing, phlegm production, wheezing, chest tightness, and nausea. On the other hand, prolonged exposure to DEPs can result in the development of asthma, worsening respiratory condition, and allergies. And additional DEPs can cause allergic reaction and fatigue, affecting both the system immune and overall well-being. With the shift towards an alternative fuel option like diesel natural gas blends and the addition of nanoparticle additives such as functionally shaped cardboard nanotubes, there is a significant potential to reduce the adverse effect of diesel exhaust particles. Natural gas, for example, burns cleaner than diesel, leading to a reduction in particulate emissions and helping to lower respiratory and cardiovascular risks associated with DEPs. Meanwhile, CNTF acts as a combustion enhancer, promoting more complete burn and reducing the release of unburned hydrocarbons and particulate matter. Together, these innovations contributed to cleaner air, improved health outcomes, and more sustainable approach to diesel combustion. Our study aims to explore how these modifications in fuel and now additives impact gene expression in human bronchial cells and identify mechanisms that could mitigate depth-induced damage. As I say, our main goal was to compare the transcriptomic responses of our A549 cells exposed to different types of diesel with CNTF, diesel natural gas, and diesel natural gas with CNTF. Through this, we aim to uncover gene expression changes and mechanisms relevant to respiratory health. Let me briefly walk you through our methodology. First, we collect depths using a cascade impactor connected to a motor generator, simulating vehicle driving cycles. Particles were captured at 2.5 micron size to reflect typical airborne depths. We then applied 4 treatments to the A549 cell, standard diesel, diesel CNTF, diesel natural gas, and diesel natural gas plus CNTF. Cells then were exposed to these particles for 48 hours, after which RNA was preserved and sent to a RNA sequencing. In our bioinformatics analysis, we processed the sequencing data through several steps: quality control, read alignment to the human genome, quantification of gene expression, and normalization using the TPM method. We identify differential expressed genes using this tube, applying a p-value cutoff of 0.05 and log for change threshold. To understand the functional implications, we use Enricher for gene ontology and KLG pathway analysis. Enricher allows us to classify genes into categories like molecular function and biological process, as well as key AGG pathways related to metabolisms and signaling. This analysis highlights functions and pathways with significant representation based on our experimental conditions. Our RNA sequencing analysis revealed that exposure to depth altered the expression of 1,226 genes overall. Here's a breakdown where there were 366 genes were impacted by diesel treatment, 513 35 by diesel CNTF, 61 by diesel natural gas, and 244 by diesel natural gas. GO and KHGG enrichment analysis show distinct patterns for each treatment. Pure diesel tests primarily affect genes involved in DNA replication, cell cycle senescence, and DNA repair. Diesel CNTF exposure activates not only these genes, but also those for nucleotide excision repair and DNA metabolism, suggesting enhanced repair mechanisms. In contrast, the diesel natural gas blend impacts genes related to DNA repair, glutathione metabolism, and autophagy, while diesel natural gas CNTF depths activated pathways in DNA replication and various DNA mechanisms, including base and mismatch repair. Our analysis showed that each deep type induces specific responses. Diesel depths alone trigger inflammation and oxidative stress, with genes like LCN2 and FOS upregulated, with DNA repair genes like LIG1 and POL were deregulated. Diesel CNTF depths enhance repair and cell cycle control, activating genes such as MCM3, MCM7, and FAM1. Then, the diesel natural gas blend emphasizes repair mechanisms regulating HSPA1B along if IEGFBP3 and the regulating cell cycle regulators like CCNND1 and E2F1, allowing cells to pause the cycle for repair. The diesel natural gas CNTF combination showed the most balanced response, activating DNA stabilizing genes like MCM4, CDC44, and RFC4, alongside stress regulators NGF and PENK. This treatment seemed to provide a protective effect with reducing inflammation and better adaptation to depth-inducing stress. In conclusion, our study suggests that the addition of CNTF to depths enhances cellular repair and adaptation, reducing inflammation and providing a more protective response compared to diesel depths alone. The diesel natural gas and CNTF combination in particular shows promise in supporting cell cycle regulation and DNA repair without triggering intense inflammatory responses. These findings open new possibilities for using functionalized nanotechnology like CNTF to mitigate respiratory health risks associated with diesel exhaust. Thank you for your attention. I'd be happy to answer any question about our study and discuss potential application of these findings in the future research.